Now that we have understood what mitochondrial DNA is and how it works inside the cell, let's move to the most important question. Why does mitochondrial DNA really matter? Mitochondrial DNA may be small, but its influence is enormous. It plays a central role in how cells produce energy, how disease develop, how we age, and even how scientists trace human evolution. In this part of the presentation, we will focus on the significance of mitochondrial DNA, why it is often called the second genome, and why life as we know it would not exist without it. As we go forward, we will see how mitochondrial DNA connects basic cell biology with real world topics like health, genetics, evolution, and modern medical therapies. Let's begin by understanding why this tiny circular genome has such a powerful impact on life. Now that we know mitochondrial DNA exists, the next question is why is it so important? Mitochondrial DNA, often called mtDNA, it plays a key role in energy production. It carries genes that help mitochondria generate ATP, the energy that powers every cell in our body. Another important feature of mitochondrial DNA is maternal inheritance. We inherit mitochondrial DNA only from our mother, which allows scientists to trace ancestry and study human evolution. Mitochondrial DNA also has a high mutation rate. While this can sometimes lead to disease, it also helps understand the evolutionary changes over time. Finally, mitochondrial DNA has great medical significance. Mutations in mitochondrial DNA can lead to genetic disorders, especially affecting organs that need a lot of energy, like your brain and muscles. So even though mitochondrial DNA is small, it plays a very important role in energy production, inheritance, evolution, and human health. Now let's focus on the first and most fundamental role of mitochondrial DNA, energy production. Every cell in our body needs energy to survive and function. This energy is required for muscle movement, brain activity, heartbeat, and even simple processes like repairing cells. Mitochondrial DNA plays a direct role in this process. It encodes key proteins which are involved in oxidative phosphorylation, which take place inside the inner membrane of mitochondria. These proteins are part of the electron transport chain. Together, they help produce ATP, which is the energy currency of the cell. So if the mitochondrial DNA is damaged or mutated, these proteins do not function efficiently. As a result of this, ATP production drops. So even if the normal nuclear DNA is normal, problems in mitochondrial DNA can make energy production inefficient. In simple terms, you can say that mitochondrial DNA helps ensure that the cell is powered, always powered, and it is ready to function. So when that gets damaged, the efficiency of this energy production becomes very less. Fine. So that's why it's very important. Now let's see what happens when mitochondrial DNA does not function properly. So mutations in mitochondrial DNA can disrupt energy production. When cells do not receive enough amount of energy, they cannot perform their normal functions. So this leads to a group of conditions known as mitochondrial diseases. Because energy demand is highest in organs like brain, muscle, heart, these organs are affected first. People who have mitochondrial DNA mutations may experience muscle weakness, neurological problems, vision loss, or seizures. Mitochondrial DNA is also closely linked to human health and aging. As we age, mutations in the mitochondrial DNA accumulate gradually, so this reduces energy efficiency in cells. This explains why mitochondrial dysfunction is associated with aging and age-related disorders. So mitochondrial DNA is not just important for producing energy, it plays a very important role in maintaining health and determining how cells respond to disease and aging. So let's see. Now let's see why and understand why mutations in mitochondrial DNA are so important in human health. Mitochondrial DNA is much more prone to mutations than nuclear DNA. There are three reasons for this. First is that mitochondrial DNA is not protected by histone proteins. Unlike nuclear DNA, it is more exposed and it's more vulnerable to damage. Second reason is that mitochondrial DNA is located very close to the electron transport chain. This region constantly produces reactive oxygen species, also called as free radicals. These free radicals are capable of destroying and damaging DNA. So that is another reason why it's more prone to damage, prone to mutation, the mitochondrial DNA. The third reason is that mitochondria have less DNA repair systems. So they lack the strong proofreading and repair mechanisms that you see in your nucleus. Because of these reasons, mutations accumulate very easily in mitochondrial DNA. As a result, the mutation rate in mitochondrial DNA is 10 to 20 times higher than that of nuclear DNA. These mutations can directly affect the energy production and lead to human disorders. So let's explore further. So we have to know two terms, that is heteroplasmy and biochemical threshold. Mitochondrial diseases behave differently from your typical genetic disorders, and this is explained with the concept called heteroplasmy. What is heteroplasmy? Heteroplasmy means that a single cell will have a mixture of normal mitochondrial DNA and mutated mitochondrial DNA. Not all mitochondria in cells are equally affected. Okay, so some will function normally, whereas the others do not. Because of this, cells can tolerate mitochondrial mutations up to a certain point. This is known as biochemical or clinical threshold. Clinical symptoms appear only when the proportion of the mutated mitochondrial DNA exceeds this particular threshold. It's usually around 60 to 90%. Below this level, cells still produce enough energy. Above it, ATP production starts dropping sharply and symptoms start appearing. This explains why mitochondrial diseases show variable severity and they affect different tissues very differently. So what actually happens when mitochondrial DNA mutation, when it crosses the threshold? What actually happens? So first thing is that energy production will drop. Cells will no longer receive enough ATP to function properly. So as a result, cells start to struggle to maintain the normal activities and eventually begin to fail, right? So the most affected organs are those that require the highest energy demand. This include brain, nucleus and heart, muscles and heart. So that's why mitochondrial disorders often present with neurological symptoms, muscle weakness, exercise intolerance or cardiac problems. In simple terms, you can say, so when mitochondrial DNA mutations reduce energy supply, the organs that need the most energy are affected. This directly links mitochondrial DNA mutations to real clinical symptoms and human diseases. Right? Now let's look at how mitochondrial DNA mutations actually show up in the human body. So when mitochondrial DNA is damaged, energy production drops. And when energy production drops, cells, especially that are highly active cells, they start to struggle. Clinically, mitochondrial disorders often affect three major systems, which are called the big three. First, it's the muscle. It leads to myopathy. People may experience muscle weakness, easy fatigue, or difficulty in performing routine activities. Second is the brain, causing encephalopathy, right? Brain dysfunction. So this can lead to seizures, cognitive decline, poor coordination, or developmental delay. Third is the nervous system. This can lead to neuropathy and hearing loss, particularly sensorineural hearing loss. In more severe cases, mitochondrial dysfunctions can also lead to metabolic imbalance and organ failure. The reason these symptoms appear together is simple. Muscles, brain, and nerves require a huge amount of energy. So when the mitochondria fail, these tissues are the first to suffer. To understand this better, let's look at a real mitochondrial disease, which is MERRF syndrome. MERRF stands for myoclonic epilepsy with ragged red fibers. This disorder is caused by a point mutation in mitochondrial DNA, most commonly in the mtTK gene, which encodes transfer RNA for lysine. Because this tRNA is defective, mitochondria cannot properly synthesize proteins which are needed for electron transport chain. As a result, ATP drops, the production drops dramatically. Clinically, patients experience sudden muscle jerks, which are called as myoclonus, along with seizures, ataxia, muscle weakness, and hearing loss. A key diagnostic feature is seen during muscle biopsy, where the abnormal mitochondria accumulate and appear as ragged red fibers. Under the microscope, this case clearly shows how a single mitochondrial DNA mutation it can disrupt the energy production and lead to widespread neurological and muscular symptoms. Now moving forward, now let's understand how mitochondrial DNA is linked to aging. As we age, mutations that are gradually they start accumulating in your mitochondrial DNA. This happens because of repeated replication errors and constant exposure to reactive oxygen species. Damaged mitochondrial DNA leads to inefficient energy production. Mitochondria they start to leak electrons, generating more free radicals, which in turn cause even more damage. So this creates a vicious cycle: less energy, more damage, and declining cellular function. Over time, this contributes towards muscle weakness, slower brain function, reduced stamina, and increased susceptibility to diseases. An easy way to understand this is like an analogy is like an old car. So mitochondria are like a car engine. So when the engine is new, it runs smoothly and efficiently. As it starts to age, it produces less power and more smoke. Similarly, aging mitochondria produce less ATP and more cellular stress. It plays a central role in the aging process. Up to now, we have seen how mitochondria affects health, disease, and aging. But mitochondrial DNA has another fascinating role as well. It helps us to understand and travel back in time. In this section, we'll see how mitochondrial DNA acts as a molecular time machine. It allows scientists to trace human origins, migrations, and evolutionary history. Because mitochondrial DNA changes slowly and predictably over generations, it preserves a record of where we came from, how populations moved, and how humans evolved. This makes mitochondrial DNA not just a molecule of energy, but a historical document which are written inside our cells. So let's see. So why is mitochondrial DNA so important for studying evolution? First, mitochondrial DNA has a high mutation rate. We have spoken about this before. These small changes they accumulate over time, allowing scientists to detect evolutionary differences even over relatively short periods. Second reason is that mitochondrial DNA follows maternal inheritance. Because it passes only from mother to child, it creates clear and simple lineage patterns without mixing from both parents. These mutations act like ticks on a clock. Each mutation marks the passage of time, helping scientists estimate when the populations diverged, sometimes within the last 50,000 years. Another advantage is the high copy number. Each cell has hundreds to thousands of mitochondria, making mitochondrial DNA much easier to recover from ancient bones or fossils than compared to nuclear DNA. So together these features make mitochondrial DNA a powerful tool for evolutionary research. So now let's look at how scientists how they actually use mitochondrial DNA in research. So one key concept is the molecular clock. Because mitochondrial DNA mutations they occur at a fairly steady state, scientists can calculate how long ago two populations or even two species they shared a common ancestor. Another important application is population genetics. So by studying groups of related mitochondrial DNA sequences, these are called as haplogroups. Scientists now can track how humans migrated across the globe using these haplogroups. So you can think of haplogroups as genetic surnames. They give clues about whether a person's ancestors they came from Africa, Europe, Asia, or other regions thousands of years ago. This is why mitochondrial DNA is widely used in evolutionary biology, anthropology, and genetic history studies. These scientific principles have very real applications. One major discovery made using mitochondrial DNA is the out of Africa theory. Mitochondrial DNA evidence showed that modern humans originated in Africa and then later spread to other continents. In forensics and archaeology, mitochondrial DNA is extremely valuable because it survives longer than nuclear DNA. It has been used to identify ancient remains, historical figures, and even victims of disasters like the Romanov royal family. Mitochondrial DNA is also used for species identification. Scientists compare the mitochondrial DNA to determine how closely related different species are or to identify cryptic species that look identical but are genetically distinct. These applications highlight how mitochondrial DNA connects biology with history, archaeology, and conservation science. It's fascinating, right? Finally, let's talk about one of the most famous ideas and exciting ideas in human genetics, that is mitochondrial Eve. What is mitochondrial Eve or who is she? Mitochondrial Eve is the name given by scientists to this one woman from whom all living humans have inherited their mitochondrial DNA. So she lived approximately 150,000 to 200,000 years ago, most likely in East Africa. It is very important to understand that she was not the only woman who lived at that time, who was alive at that time. She is simply the woman whose maternal lineage remained unbroken over generations. Other women lived alongside her, but their direct maternal lines eventually died. For example, if they only had sons or if their daughters had no children. Mitochondrial Eve represents the root of human maternal family tree, not the beginning of humanity itself. This concept beautifully shows how mitochondrial DNA links modern humans to a shared ancestral past. Fine. So she represents the root of human family tree. Fine. So this is the concept of mitochondrial Eve. It's quite important to understand. Fine. Now let's look at how mitochondrial DNA is used in forensic science. So in forensic investigation, DNA evidence is often old, damaged, or it's present in very small amounts. This is where mitochondrial DNA becomes extremely valuable. Unlike the nuclear DNA, which exists only in two copies per cell, mitochondrial DNA is present in hundreds to thousands of copies in every cell. Because of this high copy number, mitochondrial DNA can still be recovered even when nuclear DNA has completely been degraded. Mitochondrial DNA is also highly resistant to environmental damage like heat, exposure, moisture, and long-term exposure to time. So this makes it especially useful in cases involving burned remains, skeletonized bodies, or very old samples. So mitochondrial DNA gives forensic scientists a reliable genetic tool when standard DNA analysis fail. Let's now understand why mitochondrial DNA works so well in forensic identification. First, mitochondrial DNA has a high copy number. So this means that even tiny samples like a single hair shaft or a small bone fragment, they can provide useful DNA. Second is that mitochondrial DNA is more resistant to degradation, so it survives conditions that destroy nuclear DNA like fire, burial, or prolonged exposure to the environment. Third is the mitochondrial DNA follows maternal inheritance. So everyone inherits the mitochondrial DNA from their mother, and it remains nearly identical among siblings and maternal relatives. So because of this, forensic experts can identify unknown remains by comparing the mitochondrial DNA with that of a living maternal relative, even a distant one. In short, the mitochondrial DNA allows identification when no other genetic option is available. So this is also really great and fascinating. Mitochondrial DNA plays a crucial role in real-world forensic cases. One application is to identify missing persons. So mitochondrial DNA is widely used to identify victims of wars, natural disasters, mass graves, and cold cases that are decades old. It also has solved famous historical mysteries. For example, mitochondrial DNA analysis was used to confirm the remains of the Romanov family, the last Russian royal family. and King Richard III, whose skeleton was discovered underneath a parking lot in England. So another important use is hair analysis. Nuclear DNA usually requires the root of the hair, but mitochondrial DNA can be extracted directly from the hair shaft itself. These applications show that how mitochondrial DNA connects modern science with justice, history, and human identity. Now, finally, beyond forensics, mitochondrial DNA has enormous scientific and research importance. In medicine and research, mitochondrial DNA helps scientists to understand how mitochondria influence health and disease. This knowledge has opened new doors in genetics, metabolism, and cell biology. So, one of the most exciting advances is mitochondrial replacement therapy, or MRT. So this technique aims to prevent the transmission of severe mitochondrial diseases from mother to child by replacing the faulty mitochondria with a healthy one. Mitochondrial DNA research also helps scientists to study energy metabolism, aging, and how cells respond to stress. So overall, mitochondrial DNA is not just a diagnostic tool. It is a gateway to understanding life at the cellular level. Mitochondrial DNA plays a very vital role in modern medical research. In drug testing, scientists examine how the new drugs how they affect mitochondria to avoid mitochondrial toxicity, which is a common side effect of some antibiotics and antiviral medications. In cancer research, mitochondrial DNA mutations are frequently found in tumor cells. Studying these mutations help researchers understand how cancer cells reprogram their metabolism to grow and survive. Mitochondrial DNA is also very important in stem cell research. So during stem cell development, mitochondria undergoes a reset process, and understanding this helps scientists to explore tissue regeneration and future therapies. So together, these applications they show that mitochondrial DNA research is shaping the future of medicine, from safer drugs to advanced genetic therapies. So while we're discussing that, let's look at one of the most exciting and advanced medical applications which are related to mitochondrial DNA, that is mitochondrial replacement therapy, or MRT. So some mitochondrial diseases are very severe and they are passed from mother to child through mitochondrial DNA. Until recently, families carrying these mutations had very limited options. But MRT, or mitochondrial replacement therapy, offers new hope. This technique is also called as three-parent baby method, that name can be misleading. In reality, it is a carefully designed medical procedure to prevent the inheritance of faulty mitochondria, not to change who the parents are. Here's how it works in simple terms. The mother's egg has unhealthy mitochondria, but her nuclear DNA is perfectly normal. Scientists carefully they remove the nucleus from this egg and place it into a donor egg that has healthy mitochondria but not nucleus of its own. This reconstructed egg now contains nuclear DNA from the mother and father, which determines traits like appearance and personality. Healthy mitochondrial DNA from the donor, which ensures proper energy production. So after fertilization through IVF, the result you have is a healthy baby who's free from mitochondrial diseases. So MRT does not alter your identity. It simply breaks the chain of disease transmission, allowing families to have healthy children. Right? So the result is that the baby will be having a nuclear DNA from the mother and father, but it will also have a healthy mitochondrial DNA from the donor. So it cures the lineage of the disease. So you won't have a unhealthy mitochondrial. Okay, so that means you won't have the disease as well, right? So the children will be healthy, right? Now, before we conclude, let's briefly bring everything together. Mitochondrial DNA can be small, but its impact is enormous. It plays a critical and central role in energy production, keeping every cell in our body functioning properly. Mutations in mitochondrial DNA can lead to serious health disorders, especially affecting organs that need a lot of energy, like the brain, muscles, and heart. Beyond health, mitochondria helps us understand human evolution. trace ancient migrations, and uncover our shared ancestry through the concepts like mitochondrial Eve. In forensic science, mitochondrial DNA has proven to be valuable because it helps to identify missing persons, disaster victims, and even historical figures when the nuclear DNA is unavailable. And in modern medicine and research, mitochondrial DNA continues to guide new discoveries from cancer biology to regenerative medicine and advanced therapies like MRT. In short, mitochondrial DNA connects energy, health, evolution, forensics, and science, all within a single cell. Amazing and fascinating. So now, as we come to the end of this journey, it's worth pausing for one final thought. Mitochondrial DNA is more than just a genetic molecule or an organelle. It's a record of our past, a driver of our present, health, and a key to the future medical breakthroughs. From powering each heartbeat to tracing human origins across continents to help solve medical mysteries and save lives. Mitochondrial DNA quietly connects energy, evolution, health, and time all together within the microscopic space of a single cell. And that reminds us of something truly powerful. Sometimes the smallest parts of biology tell the biggest stories. Thank you everybody for thank you everyone. So we'll meet in the next video. Fine. Thank you. Please watch and share and subscribe to our channel. Thank you.